All right. So we've covered speciation, and you should have a good idea about how it basically happens, the lines of evidence that we can use for it, um, the mechanisms by which it happens, and the patterns it tends to create in space and time, like species flocks and like the patterns that we see in that become clear when we add plate tectonics to the mix. But you may still be wondering, what about new structures? I mean, it's great that one finch can give rise to 14 species of finch, or one African Rift Valley cichlid can give rise to 1,000 species of African Rift Valley cichlid. But where's the big shifts? Where are the evolution of legs or lungs or wings or bones or things like that? How do we get from you know how do we get from the goo to the zoo to you? How do we get the really big plays? So can natural selection explain the origins of new organs, new major functions, new features, stuff that's radically different? I'll start by talking about eyes. There's a human eye in cross section at the upper right. That's a regular human eye at the lower right. And one of the things that I periodically hear from people whose brains got fried by listening to too much Petra back in the 1980s is that Darwin supposedly admitted that evolution was impossible because there was no way that something as complex as the human eye could evolve by natural selection. And in fact, the quote that I usually get is this: to suppose that the eye, with all its inimitable contrivances for adjusting the focus to different distances, for admitting different amounts of light, and for the correction of spherical and chromatic aberration. Okay, we got lost. To suppose that the eye could have been formed by natural selection seems, I freely confess, absurd in the highest degree. What bothers the heck out of me is that the tracts that I used to get slipped under my door never did quote Darwin's rebuttal in the next sentence of the book. It's not surprising because the next sentence is one of those really long triple decker Victorian English sentences. Um, and it takes a bit of bushwhacking to get through it. But let's start. Reason tells me that if numerous gradations from a perfect and complex eye to one very imperfect and simple, each grade being useful to its possessor, can be shown to exist. If, further, the eye does vary ever so slightly and the variations be inherited, which is certainly the case, and if any variation or modification in the organ be ever useful to an animal under changing conditions of life, then the difficulty of believing that a perfect and complex eye could be formed by natural selection, though insuperable by our imagination, can hardly be considered real. Okay, let's make like MC Hammer and break it down. If we can meet three conditions, then there's no objection. First of all, if you can go to living critters and find structures that range from an extremely simple eye to an eye as complex as ours, all of them useful to the critters that have them. Well, first, if you can go through nature and find everything from very simple to very complex eyes. Second, if the eye varies and the variations can be inherited, which is natural selection. And third, if any variation in the organ can be useful, then the fact that you can't understand how the eye evolved does not mean it didn't. The fact that it seems absurd that the eye could have evolved by natural selection, that's not a failure of natural selection, that's a failure of you. Just because you can't see how it happened doesn't mean that it didn't. And there were some computer simulations carried out way back in 94 by Nilsson and Pelger to try to answer this. They started with a computer model of a light sensitive spot, a spot that would have a clear layer, a pigment layer for absorbing light, and a nerve layer. So it's basically a 3-layer cell sandwich. They allow the properties of every layer. So they start with this computer model. They let the computer model multiply. So they make, I think, 10 variants of this computer model, and the properties of each layer are allowed to vary randomly by up to 1% per generation. So you start with a computer model of a light sensitive spot. You create a bunch of offspring, virtual offspring of that light sensitive spot where the properties of each layer can vary. And then you select the offspring that have the greatest efficiency. And then you use those offspring to create the next generation of virtual computer models. Repeat this as necessary. And here's what they got. That's a diagram of what they started with in cross section. It's a round spot. I mean, of course, you're seeing a slice through it, so it looks like a bar. But there's a clear layer, a pigment layer, and a nerve layer. So the idea is that light is going to come in. Here's a nice little photon. It's going to pass through the pigment layer, pass through the clear layer, get absorbed in the pigment layer, and the pigment layer will pass this on to a nerve layer, and presumably it goes on to a brain or whatever. Here's the thing about an eye spot like this. There are critters, lots of them. Flatworms are the ones that come to mind that I can think of, but there are others like this. There's plenty of living things that have an eye spot that looks like this. And they can distinguish light from dark, because these nerve layers will fire more often if there are lots of photons than if there's few. What they can't do is make an image. Here's another photon coming in from a completely different angle. And oh, look, it gets bent by the pigment layer and it stimulates the nerve. Hey, here's another one. Coming in at a very low angle. And oh, look, it gets bent by the crystal layer, goes to the pigment layer, stimulates the nerve layer. Hey, here's another one. Okay, you see what I'm getting at. An eye like this can't distinguish the direction that photons are coming from. Any photon, any wave particle of light, no matter what direction it's coming from, is going to hit the same cells of the pigment layer and stimulate a nerve impulse. So eyes like this can't make an image. Living things like this have no picture of the world around them. They can know how much light or darkness there is, but that's the most they can do. So if we start with an eye like that, let every property of that eye, like the refractive index and the curvature, others, vary up to 1% in every generation. After about 800 simulated generations, the eye has gone from being flat to being a bowl. The significance of a bowl-shaped eye is that it can now detect the direction from which light is coming. So a photon coming in this way is going to hit this part, and a photon coming in from this direction is going to hit that part, right? There's no way that light coming from this direction can hit that part of the retina, right? And light coming in from this direction will hit that part of the retina. So basically what you've got is the possibility of detecting images. Eyes like this can actually start seeing pictures of their surroundings. That only took about 800 simulated generations. After about another 800 simulated generations, the eye spot started developing a lens and a cornea, and it now looks suspiciously like our own. It's got, a lens, a cornea, a retina. It's got the same basic properties that our own does, and ours are very good at perceiving images, right? We can all see pictures of the world around us. That only took about 1,600 generations, notice. That's not a very long time. I mean, it's a long time for us. Well, it's not even that long a time for us. If each human generation is 20 years, then 1,600 generations is 32,000 years, which is, hey, sweetie, well within the time frame of our own species. That's about the age of the older famous cave paintings in Europe. I love you. Have a good day. All right. That is well within the time span of fully modern humans. There were people anatomically no different from us running around 32,000 years ago, making art and making clothes and making music and wearing jewelry and things like that. Yeah, 32,000 years is not that much time. And if you envision this happening in something with a shorter generation time, like 1 year, this is extremely rapid. Okay, so on the face of it, it doesn't look like it's that hard to go from a very simple eye spot to something as complicated as our own. Remember the first condition that Darwin imposed. It's great that we can go through all of these steps, but would any of these simulated intermediate steps be useful in the real world, on the principle that an organism is not likely to evolve a structure that is completely useless to it? Are any of these intermediate steps in our simulation likely to be useful to a real organism? Well, on the left, you see those steps in the simulation. On the right, these are actual eyes found in various mollusks. That's snails and squid and things like that. And as I've mentioned, we have some mollusks, also some flatworms that have flat eye spots, pretty much like the eye spot that we started with. There's one in cross section. You can see there's a pigmented layer of cells and a layer of nerve fibers. In other mollusks, the eye spot becomes cup-shaped. This happens to be down below the chambered nautilus. The eye is bowl-shaped, and it's working like a pinhole camera. Don't know if you folks ever made pinhole cameras, but when I was a kid, I got a little single-serving cereal box and cut a hole in it, taped a piece of aluminum foil over the hole, poked a pin through the aluminum foil, and put some camera film on the other end, opposite the pinhole, and I found I was able to take pictures. Not really awesome ones, but it worked. And there are eyes of living mollusks that work on exactly the same principles. Um, there we've got, I'm not sure whose eyes in the middle, but at the bottom right, that's a diagram of the eye of a squid, which is every bit as complex as yours. It's got a cornea, an iris, a lens. Um, it is exactly comparable to yours in terms of complexity. And the point is that having half an eye is fully functional. The argument that I often get is that there's no way that the eye could evolve by natural selection because an organism can't do anything with half an eye. Half an eye is useless. Um, because all of the parts of your eye work together, right? If I took away your lens or your cornea or your retina, if I took away one part of your eye, you'd lose most or all of your eye's function. So the idea that all of these parts work together supposedly means that they could not have evolved gradually. But in fact, in plenty of living organisms, half an eye works just fine. There are plenty of critters that get along well with a retina and an eyeball, but no lens, or that get along well with just a retina, but no eyeball, no lens, no cornea, nothing else. Living critters, we can find every stage of a gradation from very simple eye spots to complex image forming eyes. In fact, there's no real way to talk about evolution of the eye because complex eyes similar to our own have evolved something like 40 separate times in the evolutionary history of animals. There's a human eye at the upper left, at the upper right, that's an octopus eye. You can see it's got all the basic parts that ours does. It has a cornea and an iris and a lens and a vitreous body and a retina. Down at the lower left, that is a cross section through the eye of a jumping spider. Um, they've got 8 of them, I think. And in jumping spiders, those eyes are unusually large. And you can see there's a lens, there is a vitreous body, that VB thing right there. That's a jelly, transparent jelly like substance. The retina you can see back there. On the right, that's an alceopid. That's a segmented worm living in the ocean. And it's got a lens and a cornea and a vitreous body and a retina and optic nerves. Complex eyes like this have evolved multiple times. It turns out they're not that difficult to evolve. This was announced about 20 odd years ago. Even box jellyfish in the Cubozoa. That's box jellyfish are a little different from your average jellyfish. They tend to have kind of squarish shapes, and they include the really, really deadly ones. These are the ones that will force beaches in Australia to be closed down for weeks if there's box jellies in the water. These are the ones where a bad sting can kill you in about a minute. This is yet another one of the reasons I am very impressed that any Australian survives to the age of 5. They've got eyes. In fact, they've got clusters of them, 4 clusters that look like this. You can see that's the lens. And the retina is easy to make out because it's pigmented. You know, if you think about it, the retina can't be transparent because if it was, light would pass through it instead of hitting it, which is what you want. So the retina's got to have that pigment layer. So all of that brown stuff that you're seeing, that's the retina surrounding a globe-shaped lens. And if you look at pictures B and C, um, B was taken in bright light, C was taken in dim light, and you can see that they've even got an iris that can decrease or increase the amount of light hitting the retina, right? Same way that your iris does, the colored part of your visible eye. Okay, this is where I'm just kicking back and imagining you all saying, ooh, ah, wow. And I found something that is even freaking weirder. There's a group of single-celled protists called dinoflagellates. We've got them in fresh water. I sometimes see them in samples from the lakes around here. They're very common in the oceans as well. They're called dinoflagellates. Many of them have chloroplasts and are photosynthetic. Some of them aren't. There is a group of them in the family Warnoviaceae. They are found only in the oceans, and they have not been well studied because we haven't yet worked out how to keep them alive and propagate them in the lab. They have to be collected wild and they don't live for very long, so they're hard to do long-term studies. But a number of single-celled protists have eye spots that can detect light. The Warnoviaceans have eyes with a cornea and a lens. That's the lens. And then a retina down there. That darkly colored zone right there, that's the retina. There's the lens sitting on top of it. The lens is labeled H over here in cross section. I forget why it is, but the retina right there is labeled R. And I have no earthly idea, neither does anybody really, what this kind of structure is doing, because being single celled, these Warnoviaceae and dinoflagellates haven't got a brain, so they're not processing images. There's no way, well, there is almost certainly no way that a dinoflagellate can be said to be seeing with structures like this when it doesn't have a brain to do the processing. So, exactly what it's used for is a really interesting question. But the fact remains that it's a single celled critter that's got structures that look just like one of these camera type eyes with a cornea, lens, and retina. Weird. Going back one, in the box jellyfish, they've actually looked at how those eyes function in species that come in close to shore. And one hypothesis that has some support is that eyes like this are very good at detecting motion. Box jellies are, in fact, predators and would need to move towards sources of motion. So eyes like this are apparently very good at that. It's also been pointed out that they seem to be useful in enabling the jelly to figure out where the shoreline is, so it doesn't come in too close to the shore and get washed up on a beach. This is a conundrum because box jellies don't have enough of a brain that you would think they would be able to perceive an image. But eyes like this can apparently be useful even if you don't have all of the image processing machinery that your brain has. You know, your brain, talk to Dr. Padberg about this if you want to, but your brain does an enormous amount of processing of the signals it's getting from its eyes to enable you to take these, these wild, crazy, colorful blotches everywhere and break them down into objects that have a distance and have a texture and things like this. It's something that we still haven't quite figured out how to get computers how to do after 40 years of trying. But eyes like this are apparently of some use, even if uh, even if the organism doesn't have brains to do the processing. So there are times when you don't need to explain the origin of complexity as anything other than good old fashioned natural selection on natural variation. This was harnessed beginning in, I think this goes back to the 1950s, with the work of a pioneering researcher in artificial intelligence named Art Samuel. Samuel was working back in the days when computers were programmed on punch cards. These, stiff paper cards, a bit longer than an index card, that had rows printed on them where holes would be punched or not punched. And feeding a stack of those into a computer was the way that you programmed it. This was also back in the day when a major university might have a computer. And if you wanted to use it, you had to book time on it. The concept of the personal computer did not as yet exist. This is the time when the head of IBM says, "I think there is a worldwide market for about 5 computers." Well, he turned out to be wrong, but still, this is the age of mainframe computing, where a computer is basically a great big instrument, and if you want to use it, you have to book time on it. Kind of like the scanning electron microscope in our department. We don't have personal ones for everybody. Samuel was doing research on artificial intelligence, and he had to book time on the computer in the middle of the night. It was the only time when there wasn't much demand. And from the early days of AI, people have tried to program computers to play games. Because probably they're really bored and kind of geeky, and B, because it's thought to be a very useful task in uh, demonstrating how computers can learn rules of logic. So Samuels has a computer program that plays legal games of checkers. He's managed to program in all of the rules. So this thing will play you a game of checkers by the standard rules. Okay, great. But to play checkers, you don't only need to know the rules, you need to know what moves to make at a particular time. Like at the very beginning of a checkers game, there's something like 7 legal moves. Maybe it's only 6. Sorry, mental imagery there. Yeah, I think there's 6 legal moves that you can make at the start of a checkers game. Your opponent can reply with 6 legal moves, and from then on, it just gets more complex. There may be positions where you might have a dozen different options. How do you pick which move to make? Well, that's the difference between playing well and playing badly, right? So Samuels had to program in rules so that the computer didn't just sit there, so the computer could decide what move to make. And one of the things that he did was program a way of weighting moves, WEIGHTING moves, where you assign each move a score and you pick the move with the highest score. So a base move weight might be 1. But if I make a move that enables me to capture, I might give that a 2. A move that enables me to crown a king in checkers might have a weight of 5, let's say. A move that leaves me open to capture on the next move might have a weight of -2, or something like that. So Samuels has this computer that has this set of numerical weights added to it. That's basically its way of evaluating moves in different situations. Here's the thing. What he did was take variants of the program, variations on the program with different weights. So let's say version A of the program, every move that ends in a capture has a weight of 2. Version B, every move that ends in a capture has a weight of 10. Well, that means version B is going to be much more likely to capture every chance it can get. Version A might not capture on every move if there's something else that might have a higher weight, that might be more important to it. So these weights basically reflect the priorities that you're giving the program. So Samuels would create a version of his program, set it to play a certain number of games against another version of its program with different weights, let them play each other on the computer, pick the one that won most of the games, create a new variant of that with slightly different weights, wash, rinse, and repeat, do this for several hundred rounds. At the end of the day, he had a program that actually beat a world champion. Now, the world champion probably was taken by surprise. The program doesn't beat world champions now, and I'm assuming that the world champion was maybe not at the top of his game because it's a computer. Who thinks those can play games? But it's still, the program is still around, and it can play a very strong game. And the point is, it's a lot better at it than Art Samuels was. Samuels himself was not that brilliant a checkers player, and the program could beat him. That is an early example of what we call an evolutionary algorithm. Um, in an evolutionary algorithm, okay, an algorithm is just a technique for solving a problem. A set of steps that you go through to get an answer to a problem. Like multiplying binomials by the FOIL method, first, outer, inner, last, that's an algorithm. Um, Detecting whether a number is divisible by 9 by seeing if the digits add up to 9, that's an algorithm. Evolutionary algorithms are not predetermined as to how you're going to do them. What you do is you have a problem, you start with a solution to the problem that may not be a very good solution, you create a bunch of random variants of that solution, so variation. You evaluate each one of the variants to see whether it's any good or not. That is selection. You take the best variant and you use that to create another round of variants. Wash, rinse, and repeat. So what Samuels was doing was letting his computer programs vary, right? And then letting them play each other to pick out the best. That's natural selection. And then using the best to generate the next round of variance. It's plain old variation plus natural selection. Samuels was one of the first to ever do that for a computer program. There's whole books on evolutionary algorithms in computer programming now. That's not really my field, but this is one of the tools that programmers can use. It's been used, at least experimentally, in drug design. If you want to do something, if you want a drug that will carry out a particular chemical function, you start with a precursor drug, you make a bunch of variants on it, pick out the one that does what you want to best, and then create a bunch of variants of that drug, test them, pick out the one that does what you want it to do the best, and keep on going. There was at least one case where the drug in question was an anticoagulant short peptide, a chain of about 10 amino acids. And they started with a chain, created a bunch of variants of that chain, picked out the one that they liked best, that worked the best, washed, rinse, and repeat. After a few generations, they had a polypeptide whose anticoagulant ability was 10 million times what they'd started with. I think I forgot to include the slide. Let me see if I did. Ah, I did. But I learned about this on a TED talk. Look this up sometime. There is a company called Unilever that makes soap flakes. You know, dry soap that you pour into, a dishwasher or something like that, or a clothes washing machine. And in the 60s, they were having difficulty making their soap flakes because the way you make soap flakes is you fire liquid soap out of a nozzle. So you've got hot liquid soap, you shoot it out of a nozzle into a cold room, and the little droplets of hot liquid soap chill and form little soap flakes before they hit the ground. And once they've hit the ground, you do, of course, pick them up and pour them into boxes and send them out to be sold. Well, the problem was that their nozzle kept clogging up, and the soap flakes were coming out of, I think the sizes were too irregular. Now, mathematically modeling how liquids flow through nozzles turns out to be an extremely difficult problem. The differential equations that we use to model fluid flow actually cannot be solved in all circumstances. In fact, there's $1 million prize being awarded for the first person who can figure out a way to do that. And this is taking place in the 60s. Supercomputers are not yet a thing. We might be able to simulate the flow of a hot, sticky fluid through a nozzle now, but they certainly couldn't do it back then. So the engineers had no solution except for somebody who got the idea to take the nozzle, make a bunch of variants on the nozzle. They'd make 10 different nozzles that looked a little like what they'd started with, but each one varied in some way. Test each one of those, pick the one that worked best, and then make 10 more variants of that. Pick the one that worked best, and then use that as the basis for making 10 more variants, and so on. And I believe it took 160 tests, but they ended up with a nozzle that works perfectly well, that doesn't clog, that produces beautiful soap flakes every time, doesn't have that clogging problem. They don't know how it works. They can't explain how it works because, as I said, the math for simulating how fluid flows through a pipe, especially if it's a sticky fluid and a pipe with a complex shape, the math is really, really gnarly, quite possibly impossible to solve, or at least nobody's been able to figure out how to solve it in all cases. So they don't know why it works, but it works. And they got that shape by, well, an evolutionary algorithm. You take a solution to a problem, that's the shape of your nozzle, let it vary, pick out the variant that works best, use that as the basis for your next generation of designs, wash, rinse, repeat, 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 and you end up with a solution. And that's the start of an answer to William Paley. Remember, Paley had written this book called Natural Theology that was gushing with incredible examples of how nature seemed designed. And clearly, this meant that there must be a designer. The thing is that just because there is design does not mean that there is a conscious designer. Natural selection is a way that systems can design themselves. Systems can adapt themselves to the situations that they're in. And all you need is plain old variation and natural selection. Basically, the entire environment and the entire heredity of an organism collectively acts as a designer. So you can, in the examples of evolutionary algorithms that I've given, it's true that people set them up, but people set them up and then design things that the people themselves could not have designed from the bottom up. Art Samuels didn't know how to write a champion checkers game because he's not that great a player. The engineers at the Unilever soap factory didn't know how to design a better nozzle. In fact, nobody really knows because the math is so hard. What they did was simply allow variation plus selection to happen, and they reached solutions that they never could have figured out starting from square one. And this is why Daniel Dennett used to refer to natural selection as the universal acid, is something that can dissolve any problem. Okay, that's actually a pretty good stopping point. So I'm going to stop the recording.